Thank you all for joining us today. We would like to spend as much time in BioFinder, so I'll share only 2 slides that will help us frame the walkthrough in a way that may explain how the content is surfaced in the application. So the life sciences content is in essence a very large knowledge graph. It includes ontologies of biochemical entities, concepts, documents, etc. The empirical data that we curate and harmonize is forming the relationships between the graph nodes. And basically, in the BioFinder user interface, you can traverse the graph in various ways and view it from different angles. If you have more complex questions and more deep questions, then the agentic tool, Newton, is another way for you to interact with the knowledge graph. So just as an illustration, let's look at this tiny subgraph that just to explain the concept. So we can see that there are various biochemical entities here, like the proteins and diseases and ligands. And we have the graph edges that represent the relationships between them. So for example, a bioactivity record may connect the ligand to a protein by providing evidence that the ligand is an inhibitor of that protein. Or another experimental record may suggest that a protein is a biomarker of a certain disease, and so on. So today we will not attempt to walk through every feature of BioFinder, but rather we will try to work through a use case of real interest, just as a way to give you a flavor of the platform. So I'll pass it on now to Valeria that will present us the scientific use case, and then we'll continue with the demonstration.
Thank you, Or, and good afternoon, everyone. Today, we're going to share with you some tips that you can use to speed up your research in medicinal chemistry by using CAS BioFinder. So these tools are going to be based on a really hot topic in drug discovery, and I'm talking about cyclic peptides. In the slide here, we see some figures coming from a CAS study that was published by CAS scientists last week in the Journal of Medicinal Chemistry. You can access this information together with other emerging trends in science, technology, and innovation by subscribing to the CAS Insights. But now, why are cyclic peptides so important in drug discovery? So first of all, around the 25% of all the FDA-approved peptide drugs are cyclic structures, where the ring usually results in a better proteolytic stability, a stronger binding to the target, and sometimes a better oral bioavailability. Now, these molecules occupy the space between small molecules and biologics, often within the beyond the rule of 5 chemical space. So they combine the specificity typical of biologic drugs with favorable drug-like properties typical of smaller molecules. Now, in the graphs here, what we see is the analysis of the entire CAS content collection, where we can see that in the last 2 decades, there has been an increase in the cyclic peptide research in both scientific publications and patent activity. In the pharmaceutical field, the strong interest for cyclic peptides is justified by the therapeutic potential of these molecules, especially in the oncology field, while other fields, for example, neurodegenerative diseases, in this case, research remain limited due to some very known challenges, for example, the blood-brain barrier penetration. A very strong example in this field is represented by cyclosporine A, that is a blue structure represented here, the blue portion here. Cyclosporine A is a microcyclic peptide of 11 amino acids that is commonly used as immunosuppressant drug, but showed neuroprotective effects. Unfortunately, it doesn't cross the BBB. So in 2024, together with my previous research group at University College London, I published a study in Science Advances where we described the synthesis of a brain-penetrant cyclosporine A analog that we see here. We have modified the structure of cyclosporine. We have introduced a CNS targeting moiety that we see here in yellow, where the delocalization of the positive charge drives the accumulation of the molecule in the brain. This molecule has been detected in the brain after 48 hours post-dose, and it is active in experimental models of multiple sclerosis. So there is clearly a potential applicability of this same technology to other microcyclic peptides to improve their CNS penetration. So now we are going live into BioFinder to extend this technology and to perform a data-driven design. Right, so we are within BioFinder now, where obviously we can start a search based on the ligands, the target itself, diseases or scaffold. But in this case, I would like to start this journey from CAS Newton, that is the new AI assistant we have within the platform. We can use CAS Newton to access in a very quick and effective way all the entire CAS content collection. In this case, I've asked the system to provide a list of cyclic peptides within with CNS relevant targets, but poor BBB penetration, because I want to use CAS Newton to find the right candidate for the extension of the technology. So here, if I scroll down, I can see the list provided by the system, the list of these cyclic peptides based on their targets in the brain. Within CAS BioFinder, everything is connected, meaning that if I start from CAS Newton, then I can quickly select the ligand or a target here by clicking the link. So I can pass easily from Newton to the ligand page or the target page. Now, if I scroll down, I can see here that there is a summary table where I can see all these compounds reported with their CNS target class, all the properties related to the poor BBB penetration of this compound that are coming with their values. And if I scroll down, I can also see some key observations provided by the system. I see here something that was really interesting from my point of view. The system is saying that DPDP is the smallest compound of the series, and actually I can confirm it here in the table. This is the molecular weight. Log BB is comparable with the one of cyclosporin A, and the system is saying that it could be potentially used as a scaffold for the design of new BBB penetrant delta opioid agonist. So if we scroll down, we see here the related literature used by the system to provide this information. The system allows us to continue searching within the same search. So in this case, I've asked the system to provide a table summarizing all key properties and features of this compound, DPDP, including primary target and secondary target, targets used, to check the selectivity, pharmacological profile, current applications, etc. If I scroll down, I can see here that the full name is this one, because actually DPDP is cyclic version of the endogenous ligand of delta opioid receptors. If I go down, I can see the primary target is delta opioid receptor, but DPDP also binds to mu and kappa opioid receptors. So here I have some information about the selectivity as well. Other information I've asked the system, for example, pharmacological profile, everything is coming with the references. So I can check every reference if I want. If I scroll down, other information I've asked. Here, something that I found really interesting is that DPDP is primarily used as a research tool right now. So it looks like a good candidate for the extension of the technology, because we can improve the BBB penetration of a molecule that could be used in the future as a real drug to treat, for example, neuropathic pain, or also other neurodegenerative diseases where actually at the moment DPDP is used only as a probe. If I scroll down, I can see the supporting references used by the system to provide this information here, and also some related literature. As we said, everything is connected. So I can click, for example, on this reference here, very recent from 2025. And the system will redirect me to the document page, where I can see in the document page, I can see the summary tab, where I can see the abstract. Here a nice picture coming from the representation coming from the document that is talking about kappa opioid receptor agonists. I could explore the pharmacology and the proteins described by this document, and I can also have a look at the ligands described by the document. Here I have my DPDPE, and I also have other ligands. Every time that I see this icon is because a ligand has been described, has been approved as a drug. So I can click on the registry number and the system will redirect me to the ligand page. Or the floor is yours.
So as Valeria clicked the link to the reference, this was the first time we landed on a node in the knowledge graph where the reference itself is one of the nodes. And here we traverse the graph from the reference to one of the ligands that is present there. So this happens to be a small molecule. Different ligands could be different types of modalities, so each will have its own features. In this case, our ligand is also a drug, so it contains also what we call drug intelligence. So this is the information about the regulatory status. It could be any molecule that went to the clinic, whether it got all the way to approval, or just tested in various phases. So you will find the information here about the status, about the indications and primary targets in the context of the clinical trials or approvals. You further down the page can find all the different descriptors from smiles to inches and various synonyms and external links for the molecule. You can also see a few of the physical properties like a number of atoms and hydrogen bond acceptors and donors, and also a few calculated ADME properties. And the reason we calculate ADME properties is obviously that many of the ligands that we extract from the literature are very, very early research entities, so they won't have all the empirical properties. So with the computed properties, we kind of create a common ground between all them so that we can compare different ligands. If we go up the page again, you may see on the right-hand side, we are calculating or predicting the LD50 value for this molecule. So this is a calculation that is using a neural network that we trained based on our toxicity data. And again, this is done for all the small molecules in the database. And this also provides you some indication of what parts of the molecule may be contributing to any toxicity effect. At the top of the page, you can see transitions to CyFinder. So if you're interested in reactions involving this molecule, in retrosynthesizing it, or in all the references that mention this compound, you can easily traverse to CyFinder. There are other tools that are available at the top of the page that we will visit in a bit. But what we wanted to show here is how this page basically lays out the different connections that this molecule makes with different entities. So the first one is the pharmacology, so the activity of this ligand against various targets. And the targets could be protein targets, they could be cell lines, they could be organisms or other biomolecules. There are various filters that you can use to refine the set based on activity, based on properties, based on the biological context of those measurements. And you can see in the visual, currently we are seeing a histogram showing how many measurements are available for different targets. You can also switch to see who are the organizations that publish those pharmacology records, just to get a picture of who are your competitors or potential collaborators. And basically the table itself displays the pharmacology data, where importantly, every data point is linked to its source, to the reference, whether it's a paper or a patent or a preprint or other types of sources. Also, every measurement has additional experimental context that you can look at the assay model. So this will provide information about the procedure, about model systems that may be used, and about other experimental conditions. Now let's continue and traverse our knowledge graph and go to the toxicology and ADME tabs. So those are also considered part of our bioactivity collection. So just different endpoints for ADME, different endpoints for toxicology. All this information is empirical data that is extracted from various sources and provided here. You can find the proteins and diseases associated with this molecule, and you can also view a few of the clinical insights, for example, the adverse events associated with the drug. And of course, this will be available only for drugs that are in use. So here you can find the FDA adverse events reports. You can see the different reports, and you can drill down to see the individual reports and then their full clinical context. So you can click one of them and view the reports and further get the statistical significance of the association between the ligand or drug and the adverse events. And again, drill down to the individual reports and see the full context. Likewise, you can see all the clinical trials in which this molecule is involved, maybe as a primary intervention or as a secondary. So you can see this information. We again provide some visuals that convey the status of the different trials, the phases of those trials. This visual is interactive, so as you click it, it will filter the table below where you can see all the information. So you can see the geographical distribution of the trials, as well as the sponsors and the year in which they are conducted. If we further go to the right, we will find the metabolites information. So here we are providing two kinds of metabolites, the known metabolites, so those are reported metabolites from the literature, and then we are trying to complete the picture of potential metabolites by providing also a few predicted metabolites based on a rule-based method that we developed. For the known metabolites, you can also click them and find information about the enzymes that may be involved in this transformation. And again, as usual, everything is hyperlinked, so you can traverse the graph to see the actual facilitator and see other things that it may be involved in. We are going to go back to the chemical space and similar ligands. But one abstraction of the data that we can look at is, for example, the scaffold of this molecule. So we can go to see the associated scaffold. We chose the Merkle scaffold as our framework for abstraction of structures. And you can see here the main scaffold and the various sites across the scaffold where different substitution, different functional groups have been reported. On the left-hand side of the page, for example, you can look at the different filters that can look at those individual sites and show you all the functional groups that have been reported in structures in the literature. All those structures have associated bioactivity data with them. So you can investigate the structure-activity relationship by applying those filters and looking at the pharmacology of those molecules. But you can also, we wanted to do something a little bit more automated, which led us to create two types of analysis. One of them is match molecular pair analysis, which we'll visit in a little bit. And the other is the free Wilson analysis, where we can basically not only look at the empirical data, but also extrapolate and predict novel structures. So back to you, Valeria.
Thank you, Or. So in the free Wilson analysis, the first thing that the system is asking is to recognize the target we want to explore, to indicate the target we want to explore. For example, in this case, I'm going to select the first one, mu-type opioid receptor, and then we have to select the cluster. That is a group of ligands that are sharing the same structure, and they differ only at these two different R group. groups. So I can select the first one, for example, and then I will see this heat map where if I zoom, I will see that there are some known ligands and other predicted ligands. So let's get rid of the predictions just for a moment. Here we see some known ligands that are coming with the P activity. So we extrapolate these data and we use them to predict the P activity for the predicted ligands that are coming with a predicted P activity. I can also modify the table to be able to see only the ISP activity, for example. So in this case, I'm looking at this predicted structure that is coming with its predicted P activity. Okay, so I think it's time now to go back to our cyclic, macrocyclic peptides. So if I go back to the literature, to the document page, I can click to the DPDPE registry number. In the ligand page, what I would like to do is to explore just quickly the chemical space of this compound. In the 2D graph, we can see, we can explore the chemical space where I can see my ligand here together with other similar ligands. In this graph that I've created, considering two properties that are, important for the BBB penetration of compounds, the molecular weight and the log BB, but we could also potentially change this type of property. If I click here, I can see that my ligand is sharing the same chemical space with our three compounds. And I can also see here the values for the properties that I've selected to create the graph, and potentially also zoom the part, the chemical space I want to explore. I can also explore the chemical space in 3D. In this case, I've added PSA as a property. So I'm going to click on these three dots because I want to have a look at the scaffold reported for DPDP. Again, here we have all these results that are ligands that are sharing the same rings and the same connections between them. I want to run the other type of SAR analysis, MMPA. In this case, as a target, I'm going to select the first, the primary target of DPDP, delta opioid receptor. And as a cluster, I'm going to select this one because I want to explore this position, that is the carboxylic acid of DPDP, because I want to understand if I can modify in the future, if it will be possible to modify this region of the molecule without modifying the binding to the target. So I'm going to select this cluster here. What we see here is an heat map that is very similar to the one of the free Wilson analysis. The difference is here, what we see is a variation of the experimental P activity reported for these two compounds that are real compounds that are coming actually with their registry number. So I can modify the table to see only the highest P activities. I can see here that the variation for the P activity reported for the methyl ester of DPDP is not significant, meaning that there is no change in the P activity if we modify the carboxylic acid. So potentially we could introduce our CNS targeting group by modifying the carboxylic acid via esterification without losing the binding to the primary target, delta opioid receptor. So I've designed this new molecule and I've run a prediction by using our predictive analytics tool that we have here within BioFinder. So we can run predictions for up to 100 ligands. In this case, I've run the prediction for both DPDP without any modification and the new compound with the new CNS targeting group here. So I've extended the chain here. So I've introduced this spacer and the CNS targeting group here. In the pharmacology table of predictive analytics, what we see is the structure that is coming with the registry number, CAS registry number, when the ligand is known. When the ligand is unknown, it's coming, I can see here, instead of the registry number, the INCHI key. I can see the target, the organism, the known P activity, when the P activity is experimental, has been reported in the literature, and the predicted P activity that is coming with a confidence score. And this is a value that is based on 5 different methods. I want to focus only on the primary target, delta opioid receptor here, because I want to check the potency of this new analog of DPDP. So here I can see the values known and predicted for the P activity of DPDP, reported for delta type opioid receptor of Homo sapiens. And here I can see the predicted value for the new analog, the new BBB penetrant analog of DPDP. So I see here that we are losing a bit of potency, but we are still in the low nanomolar range. I can also check the substantiation for these methods. For example, this is the SAS substantiation, where I clearly see here in blue. This is the simplest motif responsible of the activity at the target. So basically it's the pharmacophore. And here what I can see is that our CNS targeting moiety is clearly outside of the pharmacophore. Maybe just another one. We can have a look at the machine learning method substantiation for delta opioid receptor for of mus musculus. In this substantiation, what I can see is that the spacer here doesn't affect the binding to the target, while other atoms are clearly detrimental for the binding to the primary target. But if we are losing a bit of potency, we are also increasing, hopefully, the concentration of the molecule in the brain. So we are balancing potency and concentration. I can also click on these other 2 targets, the kappa and the mu type opioid receptor. If this is, I think this is really interesting if we want to check the selectivity of the new molecule and compare it with the DPDP before the synthetic modification. Maybe we want to focus only on Homo sapiens targets. So here, looking at these predicted P activity values, what I can see is that we are maintaining the selectivity also for the new analog of DPDP-E. So we have officially finished our data-driven design, but we could also click on one of these targets if we want to have a look, for example, at the target page. In the target page, we see several tabs here. In the summary, we see a summary of the target, a structure of the target here, and other PDB identifiers. We have them here. We could also change the PDB identifier if we want. In this case, for example, we are looking at the crystal structure. No, actually, is the EM structure of delta opioid receptor bound to deltrophin. That is another linear peptide, another agonist of delta opioid receptor that is here. And we could also have a look at the ligand target interactions if we want. We also have some predicted structures. So here we have the AlphaFold structure for this receptor. We have also other tabs, for example, the pharmacology data reported for this target, the sequences, the protein sequence, and the related mRNA sequence. And I don't know if we have time to explore maybe other tabs or...
Yeah, so this is another page showing a node in our knowledge graph, and we can briefly show maybe the disease page just to complete the picture, just showing for each entity what special feature it has. So let's pick the first disease maybe in this table, And just note that on the summary page, we can find drug intelligence information. So this is a collection of all the drugs approved or tested for this indication. And we here summarize the main mechanism of action associated with those drugs. And you can see in the page layout, again, you can find the clinical trial, the association with proteins and ligands, but also specifically for diseases, you can also find information about biomarkers and explore different gene, protein, and other types of biomarkers. So with that, we're now going to switch, and we thought we could end the part of the presentation just by giving you a little bit of flavor of a few things that are coming up our roadmap. So we've seen a couple of analytics and predictive capabilities. We saw the pharmacology prediction in predictive analytics. We saw the LD50 predictions, the metabolites. In the analytics part, we saw the matched molecular pair and free Wilson. We are adding more and more tools. For example, tools for examining selectivity of ligands against a variety of targets, or looking at the cross pharmacology mainly within families of proteins, or even the translation of bioactivity from solvents to cells. In the predictive part, we are expanding to other endpoints that probably more relevant to drug discovery, like cardiotoxicity and liver toxicity, and we are working on predicting ADME properties as well. One example for the work we're doing with the agent is we are developing a QSAR agent that really takes care end-to-end the computational research partner for you. So you start by defining your general objective, maybe working on developing a structure-activity relationship for inhibitors of a certain target. You can be very prescriptive and tell the agent exactly which models you want to develop and which methods you want to employ. But you can also have a conversation and see how the data in the platform can support various things and make a more informed decision based on that. Once you select the method, the agent itself is taking care of all the computational aspects by spinning off the jobs, keeping track of them, reporting intermediate results to you. And at the end, it obviously reports the performance of the different methods, the different models, and you can make an informed decision of which models seem to perform and fulfill your objectives. And then you can use them within the agent to predict, for example, the activity of new compounds, or you will be able to actually deploy them also to BioFinder and use them from the user interface. So with that, we conclude our presentation, and we have time for a few questions. So Jen, back to you.
There we go. Thank you. Thank you both so much for that demonstration. That was great. This is your moderator again, Jen Smither. We have a lot of good questions in the chat already, but there's still time to enter some more. So let's just start with LD50 is not always a useful toxicity endpoint. Are you working on predicting hepatotoxicity or cardiotoxicity? And what about ADME predictions?
Yeah. So the reason we started with LD50, knowing that it is not the main endpoint that is relevant to modern drug discovery, is that we had the most data for it. So it was the good test case for us to see if the methods that we're using are actually delivering good results. Now we are building the training sets that are required for other endpoints. As mentioned, we are working on liver toxicity and cardiotoxicity. And so we are expecting to deliver those capabilities within the next few months. It will be also part of the predictive analytics. So as you load your own molecule and you want to predict their activity against various targets, part of this flow would be to actually also predict their toxicity endpoints. And when we have the ADME endpoints as well, they will be included in this flow.
Thank you, Or. Here's another one. Will all new conceived compounds for which one makes predictions remain confidential, or will they become part of a general database for SciFinder?
Absolutely, any molecule that you load as a user to BioFinder is going to remain confidential, and it's not exposed to anyone else but you. And we are not using this data also to train our models, so confidentiality is absolutely paramount to this feature.
Great, thank you. How accurate is the extracted data from the literature? Is it safe to trust the data without checking the original literature?
So generally speaking, quality is our top priority. We are obviously in this day and age, we are using various automated extraction processes, but we have scientists in the loop that basically control the quality of the data that we are extracting. So yes, you can have very high confidence about the quality of the data. It doesn't mean that there are no issues ever. If you discover something as you explore BioFinder, you can always report it to us, and we will fix any issues. We are continuously also evaluating the data, making sure that the quality is not just one thing that is done prior to releasing of the data. We return to data points and validate them. So over time, we are seeing higher and higher precision of our data.
Great, thank you. I think maybe one or two more here. What requirements need to be met to access CAS BioFinder? I currently have institutional access to only CAS SciFinder, but I'm interested in using CAS BioFinder.
So there are various ways to do that. I believe that there's still an option to self-subscribe to an evaluation. You can also reach out to the person in your organization that is in touch with CAS and get an evaluation and access to BioFinder for evaluation purposes. So you can get a few weeks of experience with BioFinder and see if this is the right tool for you. Yeah.
Great. And I think we have time for one more before the end. Is this data available through an API or an MCP?
Absolutely. So you may have noticed it in one of the initial slides here, the term CAS connections. So we obviously want our data to be available where you need it and in the shape you need it. So we are making our data available in various ways. You can access it programmatically using APIs. You can use an MCP server to access it as well. We are integrating with various tools. There's also an option to do an agent-to-agent connection. So all those things are enabled through our CAS connection platform or approach. So if you are interested in this, certainly get in touch with us and we can share more information about this with you.